Our next speaker is Jared Westbrook. Actually, Jared is a director of science with the American Chestnut Foundation, whose mission is to restore this iconic tree. Jared lives in the mountains of Western North Carolina and wants to see at least the beginning of a repopulation of Appalachian forest and with blight-resistant American chestnut trees in his lifetime. Jared will talk about integration, biotech and breeding, aided by genomics, to develop disease-resistant and regionally adapted populations. So the title of his presentation this morning is Toward Disease-Resistant and Genetically Diverse American Chestnut Population. Thank you, Jared.
Good.
It's all good. It's opening, just taking a little bit. Hey, good morning, everyone. Thanks for having me. It's a beautiful place, and it's good to see all of you after 2 years of pandemic life. So a little bit about the American Chestnut Foundation, which I work for. We're a citizen science organization. We have like 5,000 volunteers that work with us, and we have orchards from Maine all the way down into Alabama. A lot of the work, we need help from university scientists to get a lot of the work done. So we also have a lot of collaborations with universities throughout the range. And primarily what we're trying to do is develop disease-resistant populations. And this is right near my house in Asheville, North Carolina. Chestnuts on top of the mountain. This is something I want to see in my lifetime. And I think we will get there.
Jared, can you use the microphone?
Yes. So many of you might know the story of the American chestnut demise, but a necrotrophic fungus, Cryphonectria parasitica, was imported from Japan into the United States at the beginning of the 1900s. So 1904 was when this pathogen was first discovered in a zoo in the New York City. And the fungus colonizes the bark, it kills and girdles the stem. And basically this fungus spread from the Northeast all the way down into like Alabama, killing 4 billion trees within the course of like 50 years. So the trees are still living today. There's hundreds of millions of them out there, but they grow as sprouts in the understory, and they resprout over and over again. Or some of the trees haven't even been infected with blight. They're just hanging out there in the shade. They haven't been infected yet, but they're like ancient seedlings. So the trees are not reproducing in self-sustaining populations. So we call them functionally extinct. They're there, but not reproducing or adapting. Chestnut blight emerged after Asian and North American chestnut species diverged. So we've recently developed a phylogeny and did a time-dated phylogeny for Castanea species. And it looks like the Eurasian species diverged from the North American species about 33 million years ago. And there's only like eight chestnuts or Castanea species. And we also sequenced, we have downloaded some genome data from Cryphonectria, which is the fungal pathogen, and Cryphonectria parasitica emerged about 13 million years ago. So the Asian species are co-evolved with blight, and the North American species were naive. So they don't have resistance to this pathogen. What's interesting is the Asian species also kind of diverged from each other after blight emerged on the scene. And so did these species gain resistance independently through independent evolutionary events, or did they arise from hybridization? These kind of questions we're still trying to work out. What we've been doing for the past 40 years, so the American Chestnut Foundation was founded in 1983. And we were founded on this idea of doing back cross breeding. So we take the Chinese chestnut, which is resistant to blight, cross it with American chestnut, a few of these surviving trees that are flowering. And then we back cross over a number of generations back to American chestnut to get trees that retain that resistance from the Chinese tree, but have the form of American chestnut. And this idea was basically, in the 80s, this geneticist from the University of Minnesota, Charles Burnham, proposed this. And it was based on the idea that there's like very simple inheritance of this resistance. And this is before the genomic era. So we're doing all this work for 40 years, assuming very simple genetic inheritance. And recently we've been able to build in some genomic tools to kind of really understand that, look into that black box of what's going on with the resistance, how complex is it genetically. And this is a typical susceptible American chestnut. You can see like this is a tree that grows in our orchards. It gets the blight and it dies back and it resprouts over and over again. So it's never like growing as a competitive tree. With some of our backcross trees in the past few years, I've been going around all over the East Coast and phenotyping trees with our volunteers. And we do have a few really nice trees. They grow like trees, single stemmed individuals. And so we do have some selections. They have intermediate levels of resistance and somewhat high levels of American chestnut ancestry, like over 50%. And with Jason, Jason's been working with us, Jason Holliday, who will give a talk a little bit later this afternoon. We've genotyped like 5,000 trees at this point. Jason's done this. And we've looked at how American chestnut ancestry relates to that resistance. And we've basically made some selections. So out of that, 5,000 or 6,000 trees that we've phenotyped and genotyped, we are selecting like 200 trees that have intermediate levels of resistance and basically, more than 60% American chestnut ancestry. I also wanted to point out, there's a few trees at the tail of this distribution that have basically 100% American chestnut ancestry, and they still have partial resistance to blight. And there are a few, like Janice was saying, there's a few, these large surviving trees that have some natural resistance to blight. And we've been using some of that material as well in our breeding program to stack the American chestnut resistance with Asian species resistance. And this is a picture of one of those trees. They're very rare. We have like a handful of them. Like they wouldn't be enough to reconstitute a species, but it's certainly something we can use for our breeding. So I've been doing some analyses looking at where in the genome the resistance resides and how many loci might be involved. And basically what I did in this analysis is looking at percent genetic variance explained in 1 megabase windows across the genome. And this is kind of similar to QTL mapping. We've also done QTL mapping, but it hammers home the point to me. It's not a smooth curve, and you see a lot of small effect loci across the genome. At maximum, an individual locus explains like 5% of the genetic variance. So we're not seeing like major, single major QTLs, and the resistance is residing all over the genome. And then we have a couple populations, a couple Chinese trees that we used as founders, and the blue and the red lines represent the different Chinese founder populations. And this graph is showing the percent genetic variance explained by locus by each of these populations. And you can see one population has, some strong effect alleles at one location, and another has, so there's population specific loci. So it looks very complex overall. When we look at, in the genome, in our backcross trees, what portions of the genome were inherited from Chinese and American chestnut, what we're seeing is that in this figure, the dark blue is like the tree inherited both alleles from American chestnut, light blue, one from Chinese, one from American, red, both from Chinese, homozygous. you can see that there's this mosaic inheritance. You don't see like 3 major regions where there were, all the trees that we selected inherited resistance in these regions. It's just that they have this mosaic of Chinese and American inheritance. And so the implication is, with complex genetic inheritance in this mosaics, we are going to get progeny, no matter what with the breeding, we're going to get progeny with intermediate and variable resistance. We're not going to like fix the resistance at like a few loci that we were expecting with backcross breeding. So I did some simulations to see like with breeding, like what can we get in terms of resistance and American chestnut ancestry. And by selecting the parents, like the 200 parents that are really good out of that pool of 5,000, we can basically improve resistance significantly by crossing those trees together. So if we were to say that Chinese chestnut resistance is 100 and American chestnut resistance is 0, right now in our base population, we're at like 15 on that scale as a mean. If we were to select the best parents, we're up to 50 just doing that. And then if we were to like do a further generation of genomic selection for the best trees out of, crossing those very best parents together and then selecting the best progeny, we would get up to like 65 on that scale. So that's pretty good for breeding. And we can definitely do better than we have been doing. But the trade-off, because the resistance is complex, is we expect that that will increase the Chinese chestnut inheritance. So if we cross the best trees together, the kids will be like 85% American chestnut. If we do further genomic selection for resistance, they're getting down to like 75% American chestnut genome. So where in this whole scheme do we're always going to be getting trees with intermediate resistance, and if we increase the resistance more, we'll be bringing in more Chinese traits. So it's not exactly clear, what the end goal is here. So what we're doing next year is doing some proof of concept. We're basically inoculating our best progeny with chestnut blight and seeing how well they do, because this is just a simulation. We need to empirically validate it. So that's kind of where we're at right now with the breeding, but that's not the only approach we're taking. There is a transgenic American chestnut that has a gene from wheat that was inserted into the American chestnut. This gene detoxifies oxalic acid that's produced by the chestnut blight fungus. And oxalic acid is like kind of a signaling molecule that basically causes the tree to go, the cells in the stem to undergo programmed cell death. So what this gene does is it detoxifies the acid, turns the acid into hydrogen peroxide and CO2, and basically reduces the damage that the fungus causes on the tree without killing the fungus. So the fungus can still live in the bark. And this was developed by Bill Powell at SUNY ESF. And you can see, in these small stem assay trials, wild type American chestnut is wilting. The trees with that oxalic acid are in the center, and our oxalate oxidase gene, they're doing pretty well. Then Chinese chestnut even is somewhat susceptible to blight with a seedling. So it looks very promising in these early trials, but of course we haven't really tested these out in the field for that long. And we're just getting to that point at this point. So our plan is to build and to combine the breeding program with the transgenic program. And basically what we're doing is breeding this gene into wild type trees. We're breeding it into some of our most blight resistant hybrids to combine the quantitative resistance with this OXO gene. And we also have another disease, Phytophthora root rot, which I'll talk about momentarily, where we have resistance inherited from Chinese chestnut that we can combine blight resistance and this root rot resistance through breeding. So we're starting with a single founder tree from New York. It's called the Darling 58 tree. And we want to get regionally adapted populations. And so to do that, we need to basically cross the single founder tree with wild type trees from across the range over multiple generations, dilute out that founder genome, bring in more of the locally adapted genome. And so we're kind of fanning out this pedigree. We're planning on doing 5 generations of outcrossing. We're already through 3 of the first 3 generations. And so we're, continuing to breed with wild type trees. And this is under permit from APHIS because these trees are not allowed to be released at this point, but maybe in the next year, they'll be approved for release so we can start to do this at a larger scale. And how we've gotten through 3 generations and, from 2016 to now is growing the progeny under high light conditions. The chestnut trees are flowering right now. Right now, all the people are running around in bucket trucks and doing breeding probably at this moment with this pollen. And we've been growing the progeny under high light, like 16-hour days. And that kind of tricks the trees into flowering in their first year. Then we can get the pollen and then, continue that breeding. And you can see some giant trees that they've, figured out how to grow these really nicely in these highlight treatments. We're using our backcross and wild type trees as parents for the transgenic outcrossing. For the past 35 years, we've been conserving these in orchards. And you can see a map of what we've conserved. A lot of our material is the backcross material. So that's, the backcross material represents an important part of the wild population. And we've also done some population structure analysis and learned that basically the American chestnut population can be divided up into 3 different regions: the far southwest of the range, the middle, and the northeast. And our volunteer state chapters have captured basically some of that variation from each of the populations. So that's a good starting point, and we need to do more conservation of wild trees, but we have these trees in the ground ready for breeding right now. So chestnut blight, as I mentioned, is not the only impediment to American chestnut restoration. We have another pathogen called Phytophthora root rot caused by Phytophthora cinnamomi, probably the world's worst plant pathogen, kills like 5,000 plant species. And American chestnut is very susceptible to this disease. And it's been limited to the southeastern United States because of extreme cold winter temperatures basically kills the pathogen in the soil. But in contrast, the chestnut blight, it kills the trees from the roots up. So nothing lives from this. So it's also an important pathogen, and as climate is warming, it's spreading north. So we need to be thinking about it as well. So the way we're combining blight resistance and root rot resistance is through a combined biotech and breeding approach. We've been screening our backcross trees for Phytophthora resistance with the U.S. Forest Service for the past 16, 17 years. We probably have a data set of 40,000 trees at this point where we have phenotypes. We screen the progeny and we see which parents are resistant. And with Jason, with the genotyping that Jason has done, we've been able to select resistant parents that have resistance to Phytophthora, and we're starting to breed the transgenic, apply transgenic pollen to those trees. And then it's pretty simple to screen for the oxalate oxidase inheritance. There's you can take a core from a nut, put it in a solution, and it changes color if the tree inherited the gene or not. So it's a very simple test, very cheap to do, no need for genomics. And then we are planting the selected trees that inherited the oxalate oxidase gene at sites with Phytophthora in the soil. And then we're further planning on intercrossing the progeny to improve Phytophthora resistance and make oxalate oxidase homozygous. So I think that we have, with the tools that we have available to us, we are within reach of producing disease resistant and regionally adapted population. But we still want to be thinking beyond, if these approaches still need to be improved upon, what are our next steps? And can quantitative resistance be engineered? This is something that I'd really like to talk about in the panel discussion and what your thoughts are on this. So one of the approaches we're taking is looking at breaking down the resistance into different pathways and biological processes, like what is the tree's susceptibility to oxalic acid? Are the trees detoxifying this acid? Is there proteins and metabolites that inhibit Cryphonectria parasitica growth in the bark? We're collaborating with Oak Ridge to figure out what these are. And we have identified a number of metabolites that are inhibitory of Prepinectria parasitica. And we can identify the individual pathways that are involved in the biosynthesis. And then lignification to block mycelial growth in the bark. So those are some pathways that we're focused on and potentially engineering. And we have some high quality American and Chinese chestnut reference genomes that are soon to be released, 4 Chinese chestnut haplotypes and 1 American chestnut haplotype to look at the, that's foundational to doing this genetic engineering. So I thank you for your attention. And as we say, it's more than just a tree. There's so many people involved in this over multiple generations. And a lot of our funding comes from private individuals with some federal funding. So I appreciate all of your support and looking forward to the discussion.